Hello everyone, this is the last video for module number 8, and there are just a few topics to talk about. And so they're shown here on this outline. One has to do with aneuploidy. First of all, just a definition of that, how aneuploidy results, and then some examples of aneuploidy. And the last topic is talking about euploidy in plants with more than diploid number of chromosomes. So, for example, triploids and tetraploids, just some unique features of those organisms. So let's get started and talk about aneuploidy first. So aneuploidy is an individual or a cell having not an even integer multiple number of chromosomes in the cell or organism. Okay, so chromosome number is not an exact multiple of the haploid number for that species. That's a better definition. So, for example, in humans, we have two full haploid sets. Haploid set is 23 chromosomes. We have 46 chromosomes, diploid. Okay, so that's a euploid condition when you have an even integer multiple number of chromosomes of the haploid set. If the haploid set is 2 chromosomes having 4, 6, 8, that would be euploid condition. Okay? Here is an example, a simple example in which there are 3 chromosomes, 1, 2, and 3. They're even color-coded. A diploid would have 2 of each chromosome. That's euploid. I'm going to ignore this category. Below this are the two most common types of aneuploids, having a non-even integer multiple of the haploid number. So one is called monosomy, in which there are 2 of each chromosome, but only 1 copy of a third. So it's monosomic for chromosome number 3 here. And the total chromosome number would be 5. That's not an even integer multiple of the haploid number, which is 3. Okay, trisomy is when 1 chromosome is present in 3 copies, and the other 2, in this case, are present in 2 copies. Okay? Total chromosome number would be 7, whereas diploid number would be 6. Okay? So euploidy is having an even multiple of the haploid number. Aneuploidy is lacking that. Okay, how about aneuploidy, and let's talk about this in humans. So for autosomes, monosomy is pretty much always a lethal condition. If you only have 1 copy of any autosome, then that's a lethal. That individual would not survive to birth. That's also true for trisomies for autosomes, with a few exceptions. And those exceptions are shown here, being trisomic for chromosome number 21, that's Down syndrome, and it's present at this frequency at birth. Okay? Being trisomic for chromosome 13 or 18 is usually lethal. But some individuals do survive to birth, and some individuals survive to usually not later than 7 or 8 years old at the oldest. So those individuals are born with severe and multiple deformities that are, shorten the lifespan greatly, usually resulting in death before birth. Down syndrome, so individuals can survive and have a pretty long lifespan, I think 50 on average for people with Down syndrome, compared to 78 or so, whatever it is now for those with euploid chromosome condition. Okay, so that's the rule of thumb. For autosomes, being monosomic, it's lethal. For being trisomic is usually lethal with a few exceptions. Now, aneuploidy for sex chromosomes, however, is tolerated much better in humans. And that's because of the process of X inactivation that we talked about before. So individuals can have 1, 3, 4, 5 copies of an X chromosome and appear relatively normal because all except for one X will be inactivated, although there are some symptoms that go along with individuals that are trisomic, tetrasomic, and pentasomic. Okay, there are also these conditions in which the individuals have a Y chromosome or multiple Ys, or a Y and multiple X's. And the same situation holds for them. There are some abnormalities, but typically viable and doesn't significantly reduce lifespan. So this slide just reiterates what I said on the previous slide. Monosomy is lethal. Why is that? That's because having only one copy of a chromosome changes the dose for all of the genes present on that chromosome from 2 to 1. Okay? And that will expose genes that are haploinsufficient. Having only one copy will result in the mutant phenotype. And it can also uncover recessive alleles, including some that may have lethal condition on the remaining chromosome. Okay? Trisomy is usually not tolerated as well, and some exceptions. So just to look at one of these, I believe it's Patau syndrome, trisomy for chromosome 13. Those individuals have 47 chromosomes total, 3 copies of 13, 2 of all the other copies. So yeah, this is Patau syndrome. This is the karyotype. And here you can see all chromosomes, all the autosomes present in 2 copies, except for chromosome number 13. So this changes the dose for all of the genes on chromosome 13 from 2 copies to 3. And these are the syndrome of conditions that accompany Patau syndrome. A really long list of deformities and that result from that syndrome, usually resulting in an early death. How does aneuploidy come about? So the main cause is, as a result of non-disjunction during meiosis, and this can be either in the male or the female, either in spermatogenesis or in oogenesis. Non-disjunction, just to remind you, is when during meiosis chromosomes fail to separate. So this can happen in meiosis 1. In meiosis 1, remember, homologous chromosomes usually separate to different cells. However, if there's non-disjunction, one cell will get both chromosomes and the other cell will get neither. This illustration is just showing what's happening for one chromosome. We're assuming all the other chromosomes are separating normally. But if there's non-disjunction for one, what will happen through meiosis 2 is that the gametes resulting will have either 2 copies of that chromosome, these 2, or no copies. If they're involved in fertilization with gametes from a normal meiosis from the mate, that will result in zygotes having 3 copies of the chromosome, or 1 copy. Monosomics would die. Trisomics would likely die as well. Aneuploidy can also result from non-disjunction in the second meiotic division. So let's say meiosis 1 went normally, each daughter cell got 1 chromosome, and meiosis 2, the sister chromatids are going to separate. That happened normally in this division, but not so in this division. In this division, one cell got both chromatids, the other cell got neither. All right, so if these gametes are involved in fertilization with a normal gamete contributing mate, here would be 2 normal zygotes. All right, here would be a zygote being trisomic, 2 copies coming from this individual, and 0 copy from the individual. This would be a monosomic individual. All right, so aneuploidy can result from non-disjunction in meiosis in either parent, male or female, and in either meiotic division, meiosis 1 or meiosis 2. Now, Down syndrome is one type of aneuploidy that can result, and this occurs in about individuals born with Down syndrome are seen in about 1 out of 700 live births. This syndrome refers to a set of symptoms that are seen, and they're listed here. Typical eye fold of the upper lid, eyelid. The face tends to be rounded and flattened in the front, relatively short, broad hands, poor muscle tone, physical and mental development is delayed, and life expectancy of about 50 years. So these are just kind of an illustration of some of the characteristics. In addition to the ones listed on the previous slide, there are a number of internal organ deformities that can occur. So this is in the digestive tract, cardiovascular system, and in the skin and the skeleton as well. Not every person with Down syndrome has all of these, but they have some subset typically. All right, how does Down syndrome result? So there are actually 2 causes for Down syndrome. The most common cause is due to non-disjunction, as we just talked about, for chromosome 21. Individuals with Down syndrome resulting from non-disjunction have 3 copies of chromosome 21 and have 47 chromosomes altogether, 1 extra chromosome 21. This is seen in 95% of cases of Down syndrome. The other type of Down syndrome is known as familial Down syndrome, and this is not due to non-disjunction, but rather to a type of translocation chromosome between chromosome 21 and chromosome 14. This is translocation is a type of Robertsonian translocation that we discussed in the previous slide. This is responsible for the remaining 5% of the cases. So let's look at this first, the familial Down syndrome, in which it's due to the Robertsonian translocation. And this results from a translocation in a carrier individual. And this carrier individual can pass this translocation down, resulting in some of his or her progeny having Down syndrome. Okay, so let's look at, first of all, just some properties of the carrier individual. The carrier individual has the translocation chromosome. It's a translocation between chromosome 14 and 21. The carrier individual has only 45 chromosomes total, but this person appears normal. So this would be the karyotype of somebody who's a carrier for this Robertsonian chromosome. They have 2 copies of all chromosomes, all the autosomes except for chromosome 14 has 1 normal chromosome and 1 translocation chromosome. Here would be the normal part of 14, and here's part of chromosome 21 added to the end of it. They only have 1 copy of the normal chromosome 21. So they essentially have 2 copies of chromosome 14, all genes, and 2 copies of chromosome 21. One is the standalone chromosome, and one is translocated onto the end of chromosome 14. All right, so even though they only have 45 chromosomes, they have essentially all the genetic material that individuals with 46 chromosomes normally have. All right, so what happens if this carrier individual has offspring? So what happens is, because this individual has 1 normal chromosome 14, 1 normal chromosome 21, and 1 translocation chromosome, these chromosomes pair like this during prophase 1 of meiosis. The chromosomes can separate in different ways. These 2 chromosomes can go to one pole and this one to the other, or these two to one pole and this one to the other, or these 2 to one pole and the translocation to another. as a result of that, you can get 6 types of gametes here. Okay, so let's look what happens if this individual, the translocation carrier, produces these gametes, and these gametes are fertilized by someone having normal gametes with 1 chromosome 14 and 1 chromosome 21. Okay, so if you have the translocation carrier produces gametes either with normal 14 and normal 21, and the mate has normal 14 and normal 21, the offspring will have 2 copies of 14 and 2 copies of 21. So they'll have a normal phenotype. If the individual passes along just the translocation chromosome and the mate passes a normal 14 and 21, this person, offspring, will be a carrier individual, similar to the parent here. And they will have the normal phenotype. If the carrier individual passes this chromosome along, 1 chromosome 14 but 2 copies of 21, then this individual will have Down syndrome. 3 copies of 21, trisomy 21. If this individual passes only the chromosome 14 along, then this individual, the offspring, would be monosomic for chromosome 21, and that's a lethal condition. If the individual passes along a translocation chromosome and a normal 14, then this would produce an offspring with 3 copies of chromosome 14. Trisomy 14 is a lethal condition. If the carrier individual passes only chromosome 21 along, then the offspring would have 2 copies of chromosome 21, but only 1 of chromosome 14. That would be lethal as well. So a carrier individual for this Robertsonian translocation will produce a number of lethal offspring, some normal offspring, and some offspring with Down syndrome. Okay, so that's how it is passed on from a carrier individual. The main type of Down syndrome, most common type, is due to non-disjunction, and that produces individuals with 3 whole copies of chromosome 21. It's 47 chromosomes in all. Now, if you analyze these individuals, you find that 3/4 of them result from non-disjunction in the mother, and only 1/4 from non-disjunction occurring in the father. Why is that? If you look at a plot of a number of Down syndrome offspring versus maternal age of the mother, what you find is that mothers giving birth in the 20s and the 30s, relatively low level, 1 in 2,000, 1 in 900, of having a child with Down syndrome. After the age of 35, there's a very rapid increase. And after the age of 50, it's the incidence is 1 in 12 children of mothers older than 50 will have Down syndrome. So that's a very huge increase in the rate. Why is that? Basically because of the biology of oogenesis. In a mother in which the oogonium is, remember, these are produced in fetal life, and they start dividing upon puberty. So if the mother is 20, these oogonium have been in prophase of meiosis with homologous chromosomes paired for about 20 years. However, if she's in her 50s, these have been in this condition for 50 years. The longer the time span, the more opportunity for homologous chromosomes that should be paired to become dispaired, I guess you could say. So disjoined, no longer together. And the higher the likelihood for non-disjunction to occur during meiosis. Okay, one last topic has to do with chromosome number that's greater than the diploid number. So this is called polyploidy, and in some organisms they have 3 times 3 copies of the haploid chromosome set, 4 copies, or even higher. This is not seen in animals, but is relatively common in plants. So this is just showing that diploid is the normal state for animals, but in plants you can have triploids, 3 copies of every chromosome, tetraploids, or even more, penta, hexa, hepta, octaploids. Now, with increased ploidy size, diploid versus tetraploid versus octaploid, that increases the size of the cell and all structures associated with the plant. The cell, the stem, the flower, the height of the plant in general, and also the fruit. If we look at 2 main types, the triploids and the tetraploids, well, what do we see? Triploids have 3 copies of all chromosomes. If a triploid undergoes meiosis, that creates a problem. How do you separate 3 chromosomes during 2 divisions, resulting in 2 daughter cells? You can't do it evenly. So what happens is you end up producing gametes that are unbalanced. They will have 2 copies of one chromosome and 1 copy of the other. That's unbalanced gametes, okay? So a triploid trying to go through pairing in prophase of meiosis runs into this problem. How can you pair 3 chromosomes? Well, you can stick them all 3 together into this structure, or you can separate into 2 that can pair and 1 that's unpaired. In either case, the result's going to be the same. One daughter cell is going to get 2 copies, and the other will get 1, right? 2 and 1. So there will be an unequal distribution of chromosomes. And if we look at this in terms of chromosomes that are indicated by letters here, let's look at an organism that has 4 chromosomes, chromosome A, B, C, and D. And during meiosis, the gametes are going to get 2 of these chromosomes and 1 of the other, right? So this is going to happen randomly for every chromosome. Gamete 1 might get 2 of the A, but only 1 of the B, and 1 of the C, and 2 of the D. Whereas gamete 2 is going to get just the opposite. All right, so both of these gametes are going to be unbalanced, having 2 of some and 1 of the other. And this is going to vary with each meiosis, but most likely gametes will be unbalanced as a result. So triploids do not produce chromosomally balanced gametes, and they do not produce viable offspring. And this has been made use of in agriculture. Triploids are sterile. They don't produce viable gametes and viable offspring, and so they do not produce seeds. And so seedless varieties of some organisms like bananas and watermelon are examples of triploid organisms that produce defective seeds and therefore no offspring. And if you look at watermelon, commercially available watermelon now, and you still can find some of this diploid watermelon that produces these dark seeds, and those can be actually planted, and you can grow a watermelon plant from those. But triploid watermelon produces these floppy white seeds. Those are the seeds that have died because they are chromosomally unbalanced. So seedless watermelon is because it's a triploid species. In tetraploids, those individuals have now 4 copies of every chromosome. And tetraploids don't have the problem that triploids have. If a tetraploid undergoes meiosis, that four chromosomes can be evenly divided into 2 sets of 2. And these can be evenly distributed between daughter cells in meiosis. That results in gametes that have 2 copies of each chromosome. They will be balanced, right? 2 copies of every chromosome. So tetraploids, unlike triploids, are not sterile. And tetraploids produce even larger fruit. than diploids. And this is commercially available grapes nowadays are tetraploids, producing much larger grapes than the older version. Here's the extreme. So this is seen for a number of species now that are used commercially to grow fruit or other crop plants. Tetraploids, alfalfa, coffee, peanuts, some apples, and some pears. And octoploids, strawberries, commercially available strawberries nowadays are octoploids, much bigger than wild strawberries, which are diploids. This is a wild strawberry I picked off my lawn, and this is an octoploid strawberry that I bought at Hannaford's. All right, so that ends video number 3, and that ends the module, and that ends the course. Hope you've enjoyed the course. See ya.